Okay, so let's move on to the next part of this chapter, that is endospore formation. Okay, so microbes, they have capacity to wrap themselves up in protective covering when the conditions are unfavorable. Okay, for example, suppose there is a drought. Okay, there's a famine, there's a drought, there's no rain. Okay, for let's say 10 years. Okay, there's a drought in a place for 10 years. So for 10 years they will live inside the spore. Okay, it will slow down its metabolic activity, cover itself in a spore, and then go for a sort of a hibernation. Okay, it will slow down all its metabolic activities and then survive for the 20 years drought. And when the conditions are favorable again the rains come, the grasses start growing. No, so again when the conditions are favorable, it will remove the spore, it will come out of the spore and start dividing and behaving as if normally. Okay, so let's we did those archaebacteria. Remember those ancient bacteria which were there since the beginning of time? So how did they survive? So maybe they did this when the conditions were very bad. They produced an endospore, okay, a very thick covering, and hid inside the endospore. Okay, so they hid only one bacteria inside the endospore. The rest were wiped out, but one endosporic bacteria, it could survive, it slowed down its metabolic activity, it covered itself properly, and then when the condition was favorable, again those ancient bacteria, they survive again. Okay, so this is why the archaebacteria, they still exist till now. Okay, so let's see what they have written. Spores are formed during unfavorable environmental conditions like desiccation and starvation. Okay, so not only if there's drought, there's no water, but if there's no nutrients, okay, no food. As the spores are formed within the cell, they are known as endospores. Okay, only one spore is formed in a bacterial cell, and on germination it will give rise to the bacterial cell. Okay, so the generation will continue. Okay, if you just make one bacterial cell to survive, the rest, at least when the conditions are favorable, the generation will continue. So these are some examples of endospore forming bacteria, some cocci, some bacillus, okay, etc., etc. Let's go directly to how it forms. Okay, then now let's see where does the endospore appear in the bacteria. Okay, so bacteria, they have this rectangular shape. No, they have this rectangular shape, or this oval shape or oblong shape, you know, depending on which species. Okay, so the spore may be oval or spherical in shape. So if this is the long shape of the bacteria, okay, this is the cell wall, cell membrane, then a spore will form inside. Okay, a spore will form inside. Okay, and you must be thinking, why is the spore so small? It's because only the important things needed, like the DNA and the RNA, only that is kept, and some few cell organelles that is necessary for survival. All those vacuoles and all, no, they will not, no point trying to save this. Okay, if suppose a comet is coming towards the earth, you have to save only the important things. No, like you cannot carry all your riches to you in the next life. No, that's what they say. You carry only your good deeds, no, so that you reach heaven, right? So something like that. So they are protecting only the things that are required for the further generation. Okay, so the position, relative size and shape remain constant in particular species. The position of the spore may be, it could be in the center, it could be near the end or towards the end. Okay, it could be central, subterminal or terminal. The size, it could be larger in diameter because it's round, okay, or it could be same size as the width of the bacterial cell. Okay. So here you can see it's either could be in the middle, subterminal, or at the tip terminal. Okay. This depends on which species. Then the structure. The endospore will consist of central protoplast. Okay, the core. Protoplast is sort of what to say. When you use one term to say both things like cytoplasm and nuclei, no, your nuclear material, that is when we use the word protoplasm. Okay, so if we say protoplast means cytoplasm also there. In the cytoplasm there are cell organelles and all, no? So and the nuclear material is also there. Okay. So the core of the protoplast, it contains the important genetic materials such as the DNA, the ribosome, the tRNA, enzymes, etc. Okay, so all the things that are required for your replication to happen in the future. Okay, when the environment is favorable, like the enzymes could be the polymerases, you know, helicases, all those. When you will learn a replication of DNA and all in your 6th semester. Okay. The core is covered by a thin membrane called a core membrane, okay, or inner membrane or germ cell membrane, from which the cell wall of the future vegetative bacterium will develop. Okay. So the membrane that is surrounding the core, that will give rise to your future cell wall. All right. It is covered by a thick layer, the cortex, and then a multi-layered, thin and a tough outer spore coat. Okay, so you have the core protoplast, then you have the core membrane, then you have the cortex, then you have the many layers of thin and tough outer spore. Okay, it could be thin, but it's very tough. Okay, very tough coat. And we can differentiate that into outer and inner coat layer. In some species, it is covered by an additional covering called exosporium or exosporium, which is apparently loose. Okay, so there is your exosporium, the spore coat, the inner membrane, the cortex, and the core. Now, looking at the formation of the endospore. The endospore formation does not take place during active phase of growth. Okay, the sporulation means when the normal conditions of life goes on, it doesn't happen. Okay, it will happen in the conditions that are unfavorable for the growth. Okay, suppose due to starvation or drying or high temperature, etc. The sporulation can be induced when there is no more sulfur, carbon, nitrogen, iron, phosphates from the culture medium. Okay, so if there are none of these macronutrients or micronutrients, the cell, it knows that it's time for me to protect myself. During sporulation, the first detectable change is the conversion of compact nucleoid into an axial chromatin filament. Remember I told you the DNA is circular, no, in bacteria. It is not just this size, okay? It's very long. If this is the cell, bacterial cell, this circular DNA is so long and so compact that it's more of like this. How do I say? Do you know that if you unwind your entire DNA, okay, if this is the earth and you are here, this is you on the earth and this is the moon, okay, this is the moon that is orbiting the earth, no? If you unwind your DNA in one cell, I'm not talking about all your cells, just in your one cell, if you unwind it, it can stretch from the earth to the moon. It is that long, but it is so microscopic, and you might have done in class 12 or with the zoology, you might have done, no, those folding, DNA folding. It is so much folded that it's become so compact that it fits inside a cell. You know, God, or I don't know, is it God or is it nature? It has designed in such a way that it's so compact. Now, obviously, there's no more tourism now, but suppose you are a tourist, no, and you should carry light, no, they always say carry only one suitcase, you know, so that it's safer for you. You can hold your passport and your money in one hand, and you can hold your suitcase in another hand. But if you have like 10-20 suitcase, which one you will protect? You know, what is important is your credit cards and your, you know, identity cards and your passport, and then try to have as less luggage as you can. So what do? You used to fold your clothes so small, no? Instead of folding your t-shirt, this is your t-shirt, no? Instead of folding it into 4 ways, like you used to fold in your cupboard like this, you will roll up your t-shirt, you roll it, then it becomes very small, it becomes this much, no? So if you roll up, you can fill one t-shirt there. But if you fold it into 4, half the suitcase is filled with only one t-shirt. But if you roll, you can fill 5 t-shirts, then you can fill 2 pairs of jeans, no? Then you can fill your shoes, your slippers, if you roll them up, and your toiletries and all. So you have compacted all your stuff in only one suitcase. So the same way, okay, the same way, this DNA, no, this circular DNA is very, very long and very, very much compact, that we can call it as a nucleoid, okay, nucleoid, okay. This very highly compact circular DNA is your nucleoid. Okay, so where was I? Okay, during sporulation, the first detectable change is a conversion of the compact nucleoid into the axial chromatin filament. Then a transverse septum is laid down towards one pole, which separates into small and large portion. Okay, the small portion with its cytoplasm and DNA forms a forespore, which later on develops into a spore. Okay. Okay, so this nucleoid will turn itself into this axial chromatin filament. Okay, then what will happen? Okay, then towards one pole we will get a transverse septum that has one large portion and one small portion. Okay. Then this small portion, okay, this has little bit of DNA, okay. Okay, this small portion has little bit DNA, a little bit cytoplasm also it has. So this small part will be your forespore, means this will be your forespore, means it's going to be your future spore, okay. Let me repeat again. Okay, you have your nucleoid, no, so much, like all extra, like junk DNA also, no, that it had, that has, what to say, a lot of junk information is there, okay, that doesn't really important during our replication, I mean during bacterial replication. Even us humans, we have a lot of junk DNA, that's why it can stretch from here to the moon, okay. All the DNA that was from our ancestors, no, see, we are Homo sapiens, we still have the DNA of our ancestors, like your Australopithecus, then your hominids, then your bipedals, no, it's all there, all the junk DNA is all there, you know, the tool making, the Bronze Age, the Copper Age, whatever information that we had, okay, it is all there in our junk DNA. But simply we do not turn them on, okay. You might have noticed that there are some freaks, no, that they have a lot of hair, right, they have that werewolf syndrome. It's nothing but somehow their junk DNA got turned on, okay, which was there in our ancestors, okay, millions and millions of years ago. So we have all this junk DNA because our body, no, it is the best computer. It has enough hard drive, okay, there's enough storage. It never throws anything, because you never know when we might have to re-evolve back into what we were, right? So our DNA, it never deletes its files. It keeps storing, okay? If it's a junk, it will keep it in the junk folder. It will not replicate, it will not express, okay? Those genes will not express. So even in this bacterial cell, this nucleoid has a lot of junk DNA, okay? So what will happen is that the nucleoid will stretch into chromatin, okay, chromatin filament, and then what happened, in one pole there will be a wall formation, and whatever important information is there in the small part of the spore, okay. So this, whatever cytoplasm is here and whatever DNA material is here, is your important one. The rest we can ignore, okay. Because here this is, let's say, a time for emergency. Let's say there's a house fire. What do you save? You might save your cat or your puppy. You know, you're not going to run in and try and save your cash or your jewelry. No, that is just stupidity. You have to save the most important things. You will be running after maybe your little brother or your little sister or your cat or dog. So you save those. Those are the only thing that's important, right? You can always build a house again. No, it might be a hut, it might be wooden or a tent, but it's enough for survival. Okay, so same here. So this small portion, which has a little bit of cytoplasm, a little bit of DNA, is the spore, and it will develop into the spore in the future. It will develop into the endospore in the future. So I think it's clear enough. Then the membrane of the larger portion will grow around the spore. Okay. So now next. Okay. So this was the transverse septum, no? Then from here, no, the rest of the cell, it will sacrifice its membrane. It will send all its membrane to cover the spore part, okay. The rest of the cell, which is the junk part, it will sacrifice its membrane and it will cover the smaller part, okay, the spore. It will cover the spore. The spore, then it will increase in size. Increase is because of the membranes, no, they are wrapping, right? If you wear like 10 sweaters, you'll be so fat, no, something like that, okay. It will become opaque, means you cannot see through it, okay, because it has so many layers, no. Like even if you wear raincoats, a transparent raincoat, if you wear 100, you cannot see anything, right? So this will become your endospore, okay. So this process takes place within 16 to 20 hours, okay. So once the cell has decided, I have tried my best for the last 5 months, I have tried to eat, whatever was there. I tried to save as much water as I can, but there's nothing left, okay. There's no more nutrients, no more materials. Then within 16 to 20 hours, sporulation will form. So the cell in which the spore will form is known as your sporangium, and for some time it will remain viable. It will still be alive, okay. I mean, it was always alive, but means it is still active. It is trying to mature itself, okay. Then the sporangium, it will release the spore, okay. It will release the autospore, okay. Remember I told you this is, let's say this is a long cell, okay, a cocoid bacteria which has a cocoid shape, okay, like a cocoon shape. So the spore formed here, okay. This was the transverse septum, the spore formed here. Then the wall's contents, okay, they covered it, the wall contents. Now this cell is known as your sporangium, okay, sporangium. Then this cell will release it will explode. It will explode and release. Then this spore, it has because it is so protected from the outside world, no? It has everything that it needs for its basic metabolic functions, okay? So it will survive many different adverse conditions, okay? Like it will, just like the archaebacteria, those ancient bacteria, they could survive volcanoes, okay? It will survive extreme heat, extreme droughts, extreme freezing. Let's say we go into a second ice age, okay? These bacteria, they will become endospores, and it can survive extreme cold, okay? So chemicals and radiations, right? So toxic chemicals, radiations, they can happen, right? Like a Chernobyl disaster, or when the Japan, what was that? I forgot the name of that. In Japan, no, I think maybe 2018, there was a nuclear reactor explosion. No, I forgot the name. It was there in the news long back. So these things can happen, no, radiation poisoning. So these bacteria, they have already prepared themselves. They know, if this happen, I will do like this, okay? Then again, when normal condition happen, it will germinate. So it will germinate, and then it will reproduce and grow, okay? So let's see how they germinate when the favorable condition happen. It will go through 3 stages: activation, initiation, and outgrowth. So activation by induction of one or more factors such as acidic pH, heat of 60 degrees Celsius, compounds containing free sulfur hydrogen group, or through abrasion, okay? So it will activate when there is a pH, okay? It can survive in acidic, means not highly acidic, but when it feels that the pH of the soil, wherever it's living, it is acidic enough for it, not too highly, not too low. Or the heat, okay, the temperature surrounding it is 60 degrees Celsius. It's no longer the volcanic temperature of 150 to 100, no? It feels that it's 60 degrees, I can activate myself, okay? Or if there's a lot of sulfur hydrogen group, means a lot of food is there, okay? Then initiation. It will, when it finds amino acids like L-arginine, adenosine, etc., means there was some food, no? Maybe some dead animal or plants. So it knows that the surrounding environment has enough food. There's enough amino acid in the medium. So the spore coat will lyse. Okay, it will activate to form an autolysin. Lyse means release, when something gets released. Okay, so this compound autolysin will destroy the wall. Remember the peptidoglycan of the cortex. Then water is taken up and calcium dipicolate is released. Okay, dipicoleic acid helps to stabilize the spore protein, and both dipicoleic acid and calcium ions provide resistance to heat. So when the spore formation happened, it will concentrate certain chemicals such as calcium and this dipicoleic acid. It will keep inside its tiny spore, because these calcium and acids like dipicoleic acid, they are resistant to heat. Okay, they protect the bacteria from getting destroyed from extreme heat, extreme freezing, etc. So when the conditions are favorable, it will release all these chemicals which helped it protect. It doesn't need them anymore. No? So it releases chemical and it will take in what it needs, that is water and the nutrients. Then the outgrowth. Okay. Then the spore wall will swell and the covering will break up. It will disintegrate, and then a germ cell will emerge. Okay. It's like a pipe. A germ tube will emerge after breaking from the spore coat, and that will behave as your vegetative cell. Okay, it will behave as the reproductive cell. Each cell produces only one endospore during unfavorable condition, and during favorable condition, it will germinate and give rise to a single bacterial cell. So the spore is a perinating organ, and the process is called perinatation rather than multiplication. Okay, perinating organ means something which can persist. Okay, so the spore is a perinating organ. Okay, now this diagram is the whole process of endospore formation. So I will just send the screenshot in the classroom because I have forgot to attach it in your note. Okay, so this is the whole process that I explained in words. Okay, so there you could see we start by the there is that the nucleoid. No, we started with the nucleoid, then it became a long axial filament, and then that wall formation happened. Okay, then what happens? The nuclear membrane is covering it. Then there's a lot of spore. It became the 4 spore. Then all those different kinds of layers form. No, the thin spiny outer layer, then a thick middle layer, etc. Then when favorable condition happened, it became released. Okay, so with this this chapter is done, and then next class we will do growth and nutrition of microbes.